Hello everyone and welcome to this video on the epigenetics. Diseases such as cancer and diabetes are usually regarded as non-heritable diseases because they are not transmitted from generation to generation through the nucleotide sequence of the DNA. Now, the very big question will be, then why is it that such diseases are generally or are usually more frequent in some families with respect or as compared to other families? With the discovery of the concept of epigenetics in the 1940s, it has been shown that these epigenetics or epigenetic factors may be responsible for such differences. Also, with the discovery of this concept of epigenetics, it has also been shown that the food that we eat, our lifestyle, the way we behave, the exercises that we carry out, the way we manage stress may influence the way that our genes are expressed. Now, the very big question will be, what is therefore epigenetics? So epigenetics is the study of traits or factors which influence gene expression without affecting the nucleotide sequence of the DNA. We know why, because if it affects the nucleotide sequence of the DNA, it will become a heritable disease. So these traits, they influence gene expression without affecting the nucleotide sequence of the DNA. And these epigenetic traits are usually regarded as non-Mendelian because they are transmitted without following a regular pattern of inheritance. An example, we have microRNAs of epigenetic factors. We have microRNAs, we have methylation, and we have acetylation. So let's start with microRNAs. So what are microRNAs? These are short non-coding RNAs of about 18 to 22 nucleotides which regulate gene expression by targeting messenger RNAs. So in order to understand this, let's look at the figure below. Excuse. So here we have our microRNA. So how do microRNAs, how do they carry out their action? Or how do they act as epigenetic factors? So let me put these lines I'm putting at the nucleotide basis of the microRNA. These are the nucleotide basis of the microRNA. So microRNAs act by binding to, using their seed region to bind to the messenger RNAs. Okay, so let me also put the nucleotide basis of the messenger RNA. So when we look at this messenger RNA, they are usually longer than the microRNAs, okay? And these lines I'm putting are also the nucleotide basis of the messenger RNA. So when microRNAs bind to this messenger RNA, two things can occur. Either the messenger RNA will be temporarily silenced or they will completely degrade. Okay, so either they are temporarily silenced by the microRNAs or they are completely degraded. So if the messenger RNA is temporarily silenced or degraded, there either cases or either case there will be no translation, and if there is no translation, no protein will be produced, and therefore there will be no protein will be produced, and therefore that gene will have been regulated. Okay, so that is how microRNAs, they regulate gene expression by binding to messenger RNA, or that is how microRNAs act as epigenetic factors. You see that they have regulated gene expression here without affecting the nucleotide sequence of the D. So here we have two other examples of epigenetic factors. We have methylation and acetylation. Methylation is the addition of a methyl group, that is the methyl group in bracket. Acetylation is the addition of acetyl group, or this is the acetyl group here in bracket. Okay, so let's see how methylation and acetylation act as epigenetic factors. Here we have our chromosome. Okay, so when the chromosome is pulled out, okay, we have regions of this region where the DNA is tightly packed together, known as the heterochromatin, and we also have regions where the DNA is loose or relaxed, or in a relaxed configuration, known as euchromatin. In this position of euchromatin, this gene expression can take place, but in this configuration of heterochromatin, there is no gene expression. Okay, and then this round stuff we see here are histones. They are proteins known as histones. Protein known as histones, while this rope-like stuff here are known as, or is known as, the DNA. Okay, so how does methylation act as an epigenetic factor? So this group, or the methylation or the addition of this group, will push the chromosome to be in this configuration of heterochromatin. And when the chromosome is in this form of heterochromatin, the DNA is tightly packed together. There will be no gene expression. But on the contrary, acetylation, which is the addition of the acetyl group, or this group, will push the chromosome to be in this configuration of euchromatin, and therefore it will favor gene expression. Okay, so why methylation inhibits gene expression, acetylation favors gene expression. So that is how methylation and acetylation and microRNAs, they act as epigenetic factors. Okay, so I hope you understood something about epigenetics. Thanks for watching, and see you next time.